Okay, DNA replication. I'll try and keep this under 20 minutes. Let's see. So there's three steps to the process of DNA replication. So remember, this is occurring during S phase of interphase, which we talked about during the cell cycle. DNA replication has essentially three steps to it. The first step we refer to as unwinding. The second step we refer to as complementary base pairing. And the third step is what we call completion of the strands. So let's start with the first step, unwinding. And if we recall to our structure conversation yesterday about DNA, so remember that DNA is this double helix, right? And consists of these two strands that are intertwined in this helical shape. And those two strands are held together by hydrogen bonds. So in order for DNA replication to occur, we have to expose these nitrogenous bases in order to create two new strands. In order to expose those those nitrogenous bases, we have to break these hydrogen bonds and separate the two strands, hence unwinding. So that kind of looks like this picture down here. For DNA synthesis to occur, the two strands must be separated. This is done through a process called unzipping. So much like unzipping the two sides of your coat, in this case, the enzyme helicase is responsible for this step. So you can see helicase here attached to the DNA strand, and in this case, it's proceeding in this direction, and as it proceeds, it pulls those two strands apart. So as we go through all of these stages, what you want to be thinking about is you want to be able to identify what the step is. So in this case, step one, we're unwinding. You want to be able to describe the events occurring, and you want to be able to identify the names of the enzymes and their function in the process. So we've got step one, unwinding. We have this enzyme helicase, which is unzipping the two strands. The helicase enzyme, remember, is essentially breaking hydrogen bonds, right, that are existing between bases on opposite strands. So we've got these hydrogen bonds occurring here in between, and that helicase breaks those bonds apart. One thing that's happening, or one term that's used, sorry about that, is that sorry, we call the process of DNA replication semi-conservative. And it's referred to as semi-conservative because by the time we're done, and remember back to the cell cycle, meiosis, mitosis, remember back to when we talked about genetics, that when we copy um, the DNA and we end up with those two sister chromatids, each of those chromatids is going to contain one of the parent strands. So you've got a parent strand here, and this is a parent strand. So as we build what are called the new daughter strands, where this would be a C and an A and an A and a T and an A and so on, as we build this strand along here, this is the new daughter strand, but it's attached essentially to the original parent strand. So because in the end, we're going to have a new DNA molecule, one side is the daughter strand and one side is the parent strand, we say that it is semi-conservative. Okay? Now, last step in this unwinding process, you can see here attached are these single-stranded binding proteins, often just abbreviated as SSBs. So pause the video if you want for a second and just think about what those single-stranded binding proteins might be doing. So these single-stranded binding proteins prevent the DNA molecule from recoiling. right? So once helicase has done its work of unzipping the molecule, they bind on to each parent strand, right? Preventing those hydrogen bonds from reforming and the DNA molecule reforming the double helix. Okay, second step, complementary base pairing of new strands. So I'll go through a little bit here and then we'll look at the visuals to remind you a little bit about this DNA structure. Remember that the two strands serving as templates go in opposite directions. One's going from the 5' to 3' and 3' to 5'. The other one is going from the 3' to 5'. And so just a reminder about that, if you remember when we talked about DNA structure, so you can see I can't touch the screen here. It's going to pop off to something else. But this nitrogenous base guanine here in purple is attached to the sugar, and the carbon where it attaches is carbon number 1. And so the sugar is numbered. All the carbons in the nucleotide are numbered, in this case starting with carbon number 1, 2, 3, 4, and then there's carbon number 5 that's just outside the ring, and that carbon number 5 links the sugar to the phosphate. So if we go up this strand on the left here, we notice that the sugars are going in the direction from 3 to 5. So again, this T is attached to carbon number 1, and then we have carbon number 2, and then down here in the bottom, carbon number 3, and then 4, and then carbon number 5 outside the ring links the sugar to the phosphate. So the sugar, phosphate backbone on this side goes 3 to 5. On the other side, we say that the strand, the sugar phosphate backbone is anti-parallel because you can see that the sugars are kind of upside down, going in the opposite direction. So we start with 3 at the top, goes to 4, 5, linking the sugar to the phosphate, and then back to 3. Phosphate joins back to the next nucleotide at the 3, and then 4, 5, and so on. So we're going in the opposite direction. So this refers to the strands as being anti-parallel, and it has an effect on how replication or complementary base pairing is going to occur. So we'll see that coming up here. Now, to get the whole process started, an enzyme called primase, and you might think of the word primer, right? Begins the process by attaching a short section of RNA nucleotides to the template strand, indicating the location for subsequent enzymes and the free-floating DNA nucleotides to begin pairing. Now, why this occurs, this primase attaching RNA nucleotides, is probably linked to evolutionary history where the first genetic material may have been RNA and DNA may have came after. We won't get into a lengthy discussion, but the long and short of it is this primase, which we can see down here, this blue section is a section of RNA nucleotides, and we can see here RNA. And all that does is allow the next enzyme, which is DNA polymerase, it allows it to attach to the parent or template strands and then begin the addition of the free-floating DNA nucleotides, which will complementary base pair through hydrogen bonding, right? With the template strand or the parent strand. Okay, so we've got first step, RNA primase comes in, attaching a few RNA nucleotides at the beginning in order to allow the DNA polymerase, this guy right here, to come in and attach and then start the DNA nucleotide base pairing. So the point about paying attention to the 5', 3' end is because DNA polymerase only works in the 5' to 3' direction. So if we look at these two strands, oops, sorry. if we look at these two strands, the top strand being 3 to 5 prime, that means that DNA replication going in this direction is going 5 prime to 3 prime. And DNA polymerase only works, right? DNA polymerase only works in that, sorry, I've highlighted the whole thing, but only works in the 5 prime to 3 prime direction. And therefore, replication in this direction is what we call is continuous. On the 3 prime to 5 prime, the newly synthesized daughter strand is referred to as the, let's erase this here, referred to as the leading strand. And the other template strand is 5 prime to 3 prime, and DNA polymerase must work in the opposite direction. So you see the RNA primer here, and then DNA polymerase must work here. So what happens is as the fork proceeds this direction, as helicase unwinds, a new strand has to start from the fork with the primer and then DNA polymerase attaching the rest here. And so we kind of have this lagging bit because it's not happening continuously. So let's watch this video here in a second and we'll sort of see exactly what's going on. But long and short, the DNA polymerase must work in the opposite direction, which means it must begin from the replication fork, thus causing replication to be discontinuous. And we refer to the newly synthesized daughter strand in this section as the lagging strand. Okay, so let's have a look at this video here and see the visual.
During DNA replication,
it's a bit automated, I apologize, but the visual is very good.
One of the two DNA strands, the leading strand, is replicated continuously, or all at once, in the 5 prime to 3 prime direction. The other strand, called the lagging strand, is replicated discontinuously, or in pieces, in the 3 prime to 5 prime direction. This is necessary because DNA polymerase, the enzyme that synthesizes a new DNA strand using a template strand, can only add nucleotides to the 3 prime end of a polynucleotide strand. Overall DNA replication is semi-discontinuous. To begin DNA replication, a helicase enzyme waits for replication. The point at which the two strands are being separated is the replication fork. The enzyme primase attaches to each of the template DNA strands. Primase synthesizes a short RNA primer on each template strand. The primers are assembled in the 5 prime to 3 prime direction. After the primer reaches a certain length, primase drops off of the template strand. A DNA polymerase molecule attaches to each DNA template strand at the 3 prime ends of the RNA primers. Each DNA polymerase begins adding DNA nucleotides to extend the primer in the 5 prime to 3 prime direction. As the replication fork opens, the DNA polymerase on the leading strand template continues to synthesize a new DNA strand. However, the DNA polymerase on the lagging strand template, which can only synthesize a new DNA strand in the 5 prime to 3 prime direction, is moving away from the replication fork. Therefore, primase must again make an RNA primer on the lagging strand template, and DNA polymerase must continue making DNA beginning at the new primer. This results in a series of short fragments, known as Okazaki fragments, on the lagging strand template. The Okazaki fragments on the lagging strand template must be connected to form a complete DNA strand. To accomplish this, a different DNA polymerase recognizes and removes the RNA primers and replaces them with DNA. However, the polymerase cannot form the bond between the DNA nucleotide at the 3 prime end of one fragment and the nucleotide at the 5 prime end of the adjacent fragment, so there is a nick in the DNA strand. The enzyme DNA ligase is needed to form this bond and eliminate the nick. The process of continuous DNA synthesis on the leading strand template and discontinuous DNA synthesis on the lagging strand template continues until the replication fork reaches either the end of the chromosome or another replication fork moving toward it in the opposite direction.
All right.
Wow,
There
look
we
at
have it.
this. 94%.
Okay. Okay. Like with many topics we've covered, right? It's going to take a couple times for you to go through it, kind of rereading things, looking at the enzymes and what they do. But after the second step of complementary base pairing, we get to the third and final step, which is just completing the strands. So by the end of step 2 and complementary base pairing, final step where we make the 2 strands complete. All right. So we've got a question here. Can anyone think of what is missing with respect to the overall DNA structure? And the individual at the end of that video kind of alluded as to what was going on. Pause the video, maybe give it some thought. So essentially the process is finished on the leading strand. If we were to consider, this helicase making its way to either the end or, like it said, to a second replication fork, but it's not completed on the lagging strand. And so the Okazaki, right, the Okazaki fragments on lagging strand are not connected. Are not connected. And so that's the enzyme ligase, which is responsible for connecting those Okazaki fragments. And I think I mentioned this yesterday that those Okazaki fragments are named after two Japanese, actually a husband-wife team who discovered that nature of the DNA replication. So connecting those Okazaki fragments. So when we look at sort of the big picture, the summary down here is meant to kind of just be brief, okay? But think back to the beginning of the video. We want to make sure that we understand what's the name of the step. So we've got unzip, complementary base pairing, complete the strands. Then we want to think of what's happening in each, right? So in step 1, DNA unzips. We want to make sure we know the enzyme responsible for that. So DNA helicase unzips the 2 strands. And there's some details in there we want to know about those single-stranded binding proteins that prevent it from, recoiling. The process is semi-conservative, that we're retaining one strand of the parent in each new DNA strand that's formed, etc., right? So we've got DNA unzips, the enzyme helicase. Then we have complementary base pairing, in which hydrogen bonds are formed between uh, the new or the formed between the parent strand and the new daughter strands via the enzyme DNA polymerase. Also keeping in mind that there's this RNA primase that has to get the system going, and that there's going to be a leading strand and a lagging strand, right? Um, and then finally, you get 2 sets of DNA double helixes as the ligase goes through attaching the sugar phosphate backbone on the lagging strand. And here's to show that process of semi-conservative replication, in that we've got our parent strands here in blue, and as those are pulled apart, we'll end up with 2 new daughter strands in red on each one. And so you could think of these as the sister chromatids that we talked about before in chromosomes, right? Sister chromatids with identical DNA sequences. So if we have here GC, we have here GC, GC. We have CG, we have CG, CG, and so on and so forth, and they'll be genetically identical. And I've got 15 seconds to spare, so you can check out the video on the course website as well under our videos, and DNA structure and replication as a good review. All right, see you next class.